Back in the beginning, many millions of years ago, life was very simple for us unicellular organisms. We had a very simple way of getting energy. We would take a string of glucose, we'd split it into a couple of pyruvate, we would get ATP in the process, and then we would turn that pyruvate back into lactate or alanine, and then through a process we would turn that back into glucose. And we had a nice little cycle here, very self-contained. And that gave us 2 ATP. And that's what we needed. There wasn't much oxygen in the air, so we couldn't use a system involving oxygen very well. And so we got by with our 2 ATP, and life was good. Then one day, we did something quite remarkable. We swallowed another living organism in a process known as endosymbiosis. That other living organism we call today a mitochondria. And even today, that is living inside our cells. Now, straight away we knew we wanted this mitochondria. And we'll look at why in just a moment. But so it didn't kill us, it just didn't replicate like a normal bacteria and invade us and destroy us. We actually dove into its DNA and we took the essential parts out of it and stuck it in our DNA, so that if it wants to multiply, to replicate, we control that with our own DNA. So we translate those genes and give it back to the mitochondria when we want the mitochondria to multiply. Now the question is, why did we go to all of this effort in order to try and keep alive inside us a mitochondria? And the answer is the mitochondria could deliver to us about 18 times more energy. Where before we were having 2 ATP and we were quite happy, now we're getting in the range of 36 to 38 ATP being generated. Now, an 18 times increase in energy is remarkable. Imagine if I said to you, I will double your energy, a 2-fold increase. Imagine how much more energy you would have going to your brain, how much smarter you would be, how much more aware you would be, how more sophisticated your consciousness would be, how much more energy you would have in your muscles, how much faster you could run, how much more you could lift. An 18 times increase in the amount of energy is absolutely remarkable. But in order to accommodate this change, some changes needed to be made. But first, what this energy allowed us to do was to form for the very first time multicellular organisms, organisms that are gathering together under a collective consciousness, under one goal, and operating. They are diversifying their functions, and they are working as a collective to stay alive. This is what mitochondria and that extra energy enabled us to do. But it came at a cost. And the cost was that mitochondria, as well as producing lots of ATP, just spat out acid everywhere. It's a massive acid producer. And acid, as you might know, is very bad for us. It damages our enzymes so they don't work properly. Without an ability to contain the acid being generated by mitochondria, we would be in very deep trouble and would be best off killing the mitochondria before it killed us. Now, acid, in its simplest form, is a proton. And the best way to deal with a proton is with oxygen. Now, at this point in our evolution, the air is starting to fill up with oxygen that's being produced by a lot of plant-like bacteria that are floating around in the oceans. They're polluting the air with oxygen. So there's plenty of oxygen becoming available to us now. So we know that if we can combine this oxygen with hydrogen, we can form water. And water is pH neutral. So we've taken something incredibly acidic and we've turned it into pH neutral just by adding oxygen to it. Now, it's not as easy as just adding oxygen. We need to get that oxygen to it, to the area where the mitochondria are working very hard. To do that, we require a circulatory system. The other thing is the area that is using all this energy is also producing another substance called carbon dioxide. Carbon dioxide will naturally combine with water to form carbonic acid, which will naturally dissolve away into a bicarbonate ion and a proton. And so carbon dioxide also generates acid, as well as a bicarbonate ion, which is a buffer. Now, a buffer is something that is able to withstand acid changes in the body. Now, our body wants to be able to cash in on this as best it can. So it's going to try and use the circulatory system to compensate for this area where there's massive acidity starting to occur, such as may happen in an exercising muscle. Pause. Welcome back. So we're going to use the circulatory system to try and solve an acid problem in our tissues. This is so important that it is going to become one of the primary functions of the circulatory system. We're going to try and solve this problem by delivering oxygen to the tissues and removing carbon dioxide, which, as we saw, can produce hydrogen ions, which is an acid, as well as producing a bicarbonate ion, which is a buffer. So it should be a simple enough process to circulate the blood into the lungs. And being there more oxygen in the air than in the blood, oxygen should diffuse in. Being that there's more carbon dioxide inside the blood than in the air, oxygen should diffuse out, and our problem should be solved without any further intervention. Except for one problem, and that is that gases do not like to dissolve in fluid, and our blood is fluid. If you have a stagnant pond, then the oxygen will gradually dissolve out of it, and stagnant ponds become acidic because the oxygen part of H2O is leaving you only with your hydrogen, your acid. Similarly, if I open a can of soft drink, then all them carbon dioxide molecules are just going to start screaming up towards the surface in order to get out of that fluid. So suggesting that we can just transport oxygen around the body to the tissues or do similarly with carbon dioxide is not feasible. So what the body did was it invented a molecule that it thought could solve both those problems. It's a molecule that has a protein portion to it and has an ion in the center of it, or 4 ions actually, and that molecule is hemoglobin. Now, because oxygen has a negative charge, and because iron has a positive charge, stop. Now, because oxygen has a negative charge and iron has a positive charge, the two will attract each other like magnets. And this is enough to draw oxygen across the membrane into the fluid of the blood in order to bind very loosely with iron. And it's important we maintain a loose connection. Otherwise, we're going to have all sorts of problems getting oxygen off at the other end. Now, oxygen also has an important relationship in hemoglobin with hydrogen. It's almost a seesaw relationship. When oxygen jumps on, hydrogen is booted off, hydrogen that was on the hemoglobin. And we'll see where that came from and why that's important a bit later. So oxygen is on. When one oxygen binds, it makes it easier for the second oxygen to bind, which makes it easier for the third oxygen to bind, which makes it easier for the fourth oxygen to bind. This is called cooperative binding. So a hemoglobin molecule has cooperative binding and can bind 4 oxygen molecules. Now, by itself, that doesn't sound like such a feat. Is 4 oxygen molecules going to be enough to cater for the acid problem that is occurring in our tissues? Remember that mitochondria is just spewing out acid, as well as providing us with 18 times more energy than we've ever had before. Well, at first glance, it may seem like it's not enough. But you have to realize that the cell that is going to carry hemoglobin, the erythrocyte or the red blood cell, actually has about 250 million hemoglobin molecules on it. So now, rather than carrying 4 oxygen molecules, it's carrying in the order of 1 billion oxygen molecules from the lungs to the tissues. That's 1 erythrocyte, 1 red blood cell. How many red blood cells do we have in the body? In the order of 20 to 30 trillion red blood cells. So now we're looking at an order of 20 to 30 trillion times 1 billion oxygen molecules being transported from the lungs to the tissues in order to soak up that acid and turn it into water. That's the only reason we need to breathe oxygen, is to create water. So when the blood cell gets around, the red blood cell gets around to the area of the tissues, it encounters a slightly different environment. It's a little bit warmer. Local temperature is higher because of all the energetics that are happening within the cells of the body at this area. And that helps oxygen to jump off. A higher temperature is more Brownian motion, and that loose connection with the iron is easy to break and easy to shake. Not only that, but at the tissues we have a very high concentration, as you know, of protons, of hydrogen ions themselves. And some of that is going to bind to hemoglobin, which is going to, in that seesaw effect we talk about, help kick off oxygen. They cannot be both on there at the same time. And we'll look at where these protons come from in just a moment. Not only that, we have a very high concentration in the tissues of carbon dioxide. And carbon dioxide, as we saw, will naturally combine with water, which is what the oxygen is forming, to form carbonic acid, which will dissociate into bicarbonate ions and protons. And we saw protons are the acid, bicarbonate ions are a buffer. They kind of cancel each other out a bit. Maybe it even becomes more acidic. I'm not sure on that one. But the red blood cell, the erythrocyte, is going to capitalize on this little reaction. And inside it, it's got an enzyme called carbonic anhydrase. And it's going to facilitate this reaction, allowing it to occur at much, much faster rates than it normally does. Just let me write the equation on the board for you. Water combines with CO2. To form what we call carbonic acid. hydrase is going to facilitate that reversible reaction. And it's important to note that it is reversible depending on the concentrations. And carbonic acid will naturally split into protons, which are acid, and into bicarbonate ions, which are buffers. Okay, so it's kind of like breaking eggs. As the red blood cell comes along, carbon dioxide starts piling onto the red blood cell. And red blood cell, like cracking eggs, is breaking that down, and it's producing protons, so hydrogen ions, and it's producing bicarbonate ions. It's producing an acid, and it's producing a buffer. Now, it binds the acid inside it. So the acid can't escape back out. So the acid is producing this reaction, it's holding. It's not acidifying the blood through this reaction. And it's doing something else as well. The bicarbonate ion, being a buffer, is sent from the cell back out into the blood in exchange for chloride coming in. That's called the chloride shift. Because the body wants that buffer that's being made out in the blood in order to resist the changes caused by the hydrogen ions, because the mitochondria is spewing out heaps of acid. So not only are we using oxygen to bind with the acid, but we're also using bicarbonate ions to buffer any further changes. I should say there's one further element that helps oxygen to offload, and that's 2,3-BPG, which is biphosphodiesterase, and that just binds to some of the sites that oxygen was on and helps a conformational change to occur in the heme and helps bounce off oxygen. But that's going back a little bit. Okay, now how does carbon dioxide get back to the lungs? It gets back, some of it will dissolve into the blood, some of it will, better than oxygen. About 7 or 8% can dissolve into the plasma. Doesn't want to be there, but some will go there simply because of the high concentration we've got. About 10% will bind as carbon dioxide to that protein group we talked about in the hemoglobin. And that's called carbaminohemoglobin. It'll return to the lungs bound. And the rest of it will return to the lungs as bound protons and bicarbonate ions, which are circulating around in the blood. Okay, so we've pretty much solved the problem so far. As the blood moves now, loaded up with the carbon dioxide, surrounded by the bicarbonate ions, carrying the chloride and carrying the protons, it reaches the lung, where oxygen is waiting to come in and bind to the ion. Now the pressure, the partial pressure, which is the pressure of a single gas, the partial pressure for carbon dioxide is out of the body, into the lung, and then out into the air. Now, because of that, and because of the oxygen wanting to come in and push off those hydrogen ions, hydrogen is going to start entering the blood. That's going to combine with the buffer that we've got. So we've got hydrogen coming in now and combining with our bicarbonate ion, which is forming now our carbonic acid. The reaction is starting to reverse, and the carbonic acid is going to dissociate once again into water, which is pH neutral, and into carbon dioxide, which will breathe out. So it's a very tight little relationship here. Now when this is formed, the chloride that had been inside the cell is once again kicked out into the blood. So you often find lower chloride levels between the side of the tissue that's doing all the work and the lung, rather than on the arterial side. And I think that about covers it.